Hello, everyone, and welcome to today's webinar titled Insights for Optimizing RNA Electrophoresis Workflows. This webinar is a part of the 13th Annual Precision Medicine, Genomics, Genetics, and Molecular Diagnostics virtual event series. I am Melanie Chen of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Thermo Fisher Scientific. For more information about Thermo Fisher Scientific, go to www.thermofisher.com. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You will also notice that the speaker has provided a survey located to the left of your screen. This survey will be available during both the live session and the on-demand period. In addition to the survey, we encourage you to participate in 2 poll questions throughout the speaker presentation. Simply click on the answer when the poll questions are displayed. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar or from the lobby within the event. Now, let's get started. I now present today's speaker, Paulius Palaima, Product Manager at Thermo Fisher Scientific. For a complete biography on our speaker, please visit the Presenter tab from the menu on the left of your screen. Paulius, you may now begin your presentation.
Thank you very much for having me. My name is Paulius Palaima, and I'm a product manager for nucleic acid electrophoresis portfolio at Thermo Fisher Scientific. In recent years, RNA analysis has become an increasingly relevant topic, and in this short webinar, I'm going to talk about the most basic form of nucleic acid analysis, which is electrophoresis. I will share with you some insights for optimizing specifically RNA electrophoresis workflow. So we will cover several topics here. So we will cover the general nucleic acid electrophoresis as an introduction. So we have a baseline, and then we'll go through the RNA-specific electrophoresis workflow, covering the sample preparation, gel and buffer preparation, as well as gel run and visualization. And we will proceed with the simplification steps available for RNA electrophoresis, and finally some conclusions and resources. So, nucleic acid analysis, or through electrophoresis, is a very, very old method. It hasn't really changed in principle for over 50 years, apart from minor tweaks here and there regarding the separation matrix. And generally, it is separated into several distinct steps, meaning we have the gel preparation together with the buffer. Then we have the sample loading and separation step, in which you get your actual electrophoretic separation of your nucleic acids. Then you have the staining step if you don't have the stain inside your gel already, and then imaging and export and analysis. So the first part of this process, if you are making everything yourself, is fairly tedious, as generally speaking, it has very little hands-on time, but you have to wait for very long periods of time for either the agarose gel to set or for the actual separation of your sample. So this can take up to 120 minutes, so up to 2 hours basically of just waiting. On the other hand, we have some innovations within the electrophoresis field. So this would be namely the Invirgin e-gel electrophoresis system. And once you have prepared your sample, it's basically 3 very simple steps. You load the sample, you run the sample, and you analyze it all on one instrument without the need of preparation of all the buffers and the gels. And it just simplifies it to a process that takes about 18 minutes. So for the traditional electrophoresis, the instrumentation that is needed is also quite numerous, per se. For gel preparation alone, you need the microwave, you need the gel tank, you need a stirrer, so your agarose doesn't set just in your Erlenmeyer flask. Well, then you need a power supply to actually create the electrical fields in which help you separate your nucleic acids. And finally, you have to have an imager in this case. So within the e-gel electrophoresis system, this is all simplified into one instrument that is about the size of a toaster that you can easily store on your lab bench. Then the second part is the reagents for the traditional nucleic acid electrophoresis. You have to have the agarose, the stain, and all the different buffers, including the water that you would be using to dilute them, and that all is also simplified into one reagent that doesn't really need any additional buffering. It is dry, precast electrophoresis, and everything is contained within that one gel. So significantly simplifying the traditional, say, workflow.
We would appreciate your participation by answering a few polling questions. To record your answers, click on the option on the screen. Our first polling question asks, would you like to get a quote on electrophoresis instruments to purchase? Option A, yes. Option B, no. I will give you a few moments to select your answer. Paulius?
So let's get back to where our RNA electrophoresis is used. So generally speaking, within the context of the field of nucleic acid analysis, there are two primary functions for nucleic acid electrophoresis. So one is quality control. So this would be basically assessing your RNA integrity, checking whether the synthesized fragment is of the correct size. So all of these are steps within your workflow that are purely quality control related. And then we have the RNA electrophoresis steps that are generally associated with other downstream techniques, say northern blotting, that are more inclined with the detection of a specific transcript of a specific RNA species, potentially, or even verifying the presence of a transcript that you are searching for. And in itself, RNA electrophoresis is very much similar to DNA electrophoresis. So you still have to have a buffered you still have to have a buffer, you still have to have a gel, a matrix in which you are separating. So usually the gel matrix is still agarose, like in DNA electrophoresis. However, there are several key differences, and those key differences boil down to the two primary risks for RNA electrophoresis workflow, meaning you have to avoid your RNA from degrading, so you have to protect your RNA from degradation. And the second one is you have to have accurate results. So you have to denature your RNA because it tends to form secondary structures. So in both cases, you tend to use quite harmful chemicals to prevent this. And then like some of these harmful chemicals can be something like formaldehyde or bleach. And the whole RNA electrophoresis workflow within the context of your experiment should be performed under RNase-free conditions as much as possible. So what that means is that from extraction all the way to imaging, you have to minimize the exposure of your sample to any RNases that are present in the environment. So we will not be covering within this webinar the extraction part of sample preparation, let's say, but we will be going into just this pure sample preparation prior to loading it on the gel, and then we will go through the documentation systems as well. So to prepare your sample, there are multiple different ways. So after the extraction, obviously try to keep everything on ice to just limit the activity of your RNases and prevent general degradation of your RNA sample. And you can use either self-made buffers for the denaturation and loading to the agarose gel, or you can use something that is already available, say Thermo Scientific 2x RNA loading dye, which already has denaturing agents included. So the second step is the actual heating process of your RNA sample, and this differs depending on which temperature you're using and how long you want to heat. So say in our experience, 70 degrees for 5 minutes has been very optimal condition for RNA denaturation. And after this, just make sure that you don't load it back on ice. Just put it next to, just put it next to where you would be loading the gel, and then as fast as possible load it into the gel to prepare your electrophoresis run.
Our second and final question asks, would you like to be contacted by a sales team to make a purchase of one of our electrophoresis systems? Option A, yes. Option B, no. Please take a few moments to select your answer. Thank you so much for participating in the polling questions. Paulius, back to you.
Now, the gel itself is the part that has these like harmful chemicals. So because it's in the presence, like it usually tends to have either bleach or formaldehyde in its composition. So I chose this particular recipe because I'm fairly familiar with it. It's better to work within a fume cupboard. So for gel preparation, again, there are several different ways that you can prepare your gel, but as I'm familiar with this particular recipe, I chose the agarose, deep sea water, mops, and formaldehyde formulation. I didn't write down the specific quantities of each of the reagents that would be needed for this gel, because it all really depends on how big your gel is and for how large your fragment is. Generally speaking, the gel needs to settle for about 1 hour. So again, just to make sure that everything has to be done in the fume cupboard, so you limit your exposure to formaldehyde. The buffer that I am used to using, it's 10 times MOPS and 1 times Deep Sea treated water. So again, we use the Deep Sea treated water to limit the exposure of your sample to RNases. And overall, again, I would just very much like to stress that when possible, just please either treat all of your flasks or all of your working environments with RNA ZAP, or make sure that you work in an environment that you have already been dedicated to RNAs to just limit the exposure and limit the chance that your RNA sample will be degraded. Finally, within the traditional workflow, before starting your run, you also have to decide on what is the RNA marker that you would like to use, because you cannot use a DNA marker because DNA markers are double-stranded and they migrate very differently from the RNA markers. In this sense, we have a plethora available on Thermo Fisher Scientific, and ribolyl rulers span a wide range of sizes, so you can definitely make sure that you choose the one that works best within the context of your fragment, as well as within the context of the agarose percentage that you are using to separate your fragment, because you want the full separation range to obtain the best results. Additionally, if you are imaging your gel after the run, you have to use the stain that would bind to your single-stranded molecule. And in this case, it would be something like cyber gold, which shows a strong affinity for also single-stranded molecules. And finally, when you are working with electrophoresis, it's important to set the voltage and the buffer. Well, it's important to set the voltage correctly to make sure that you have your full separation throughout the full range and a reasonable amount of time to obtain the optimum resolution. Because if you run it too slow, it might be the sample might diffuse and degrade, and if you run it too fast, the agarose can melt and all of your results might be a bit distorted due to the high voltage present. So please keep that in mind. So how has the workflow evolved and how can it be simplified? As mentioned, I'm not going to cover the extraction because extraction protocols really depend on your end application and what is your sample. The sample preparation is really not that big of a time consumer, because we can prepare your sample in like at least 5 minutes, depending on your denaturation conditions. But this analysis and imaging workflows where you have to actually either prepare your gel and then run it is the big time consumers within all of the electrophoresis workflows. So in this case, this is where we are very lucky that eGel EX agarose gels can actually be applied to analyze RNA samples in a very, very fast manner, much faster than the traditional methods. So we have two of them available. So one is the eGel agarose a gel with 11 wells, and the other one is with the 22 wells for very fast analysis. Both of them can be run on either the eGel PowerSnap Plus or the smaller eGel PowerSnap systems. So how does it compare? Generally speaking, I'm sure that you probably recognize the right side of this slide, because I showed that at the very beginning, because it still applies. So generally speaking, with the traditional RNAs workflow, we have about 60 minutes for the gel preparation, including all the sample preparation, which is just, some hands-on work, but just a lot of time just waiting for the gel to set. Then you have the RNA sample preparation with the gel run, which is also an additional 80 minutes. So generally speaking, like all of this, apart from the sample preparation, can be condensed significantly. And even in the case of using the PowerSnap systems, you can even condense the time needed for imaging, export, and analysis as well. So the full workflow, apart from the sample preparation, can be very much easily condensed into about 18 minutes, depending on which agarose gel you're using on the eGel electrophoresis system. So some of the key takeaways here is protect your sample from degradation. So I think I made it significantly clear throughout this presentation that this is the key moment. Denature your RNA prior to loading for best results. So make sure that you have the correct size of your fragment when you're analyzing it. You have to select the right ladder for your application to make sure that you size it correctly for your experiment. Use the proper staining dye, because otherwise, if you want to image the gel after your run, you won't be able to see it. There are additional little tidbits that can improve your analysis. So use wider comps for better view. This just improves the sharpness of your image, of your band, and also use lower voltage for best results. So previously in one of the slides, I wrote about 1 hour at 60 volts. That tends to be quite optimal, but in some cases, you can go lower, just don't go too low, because otherwise your sample will just diffuse and degrade. Some things that you would like to avoid, so contaminating your RNA samples with RNAs, which will just degrade your samples, overloading the sample as well, damaging the wells when you are loading. This applies to your homemade agarose gel as well as the eGel EX gels. Improper amount of buffer in the electrophoresis tank, so just make sure that everything is submerged correctly. And finally, never give up, because your science matters. So for additional information on the available reagents for your RNA electrophoresis workflow, please go to our Molecular Biology Education Library, as well as the websites associated with the products and the electrophoresis workflow. Thank you very much.
Thank you again, Paulius Palaima, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today. I'd also like to thank our sponsor, Thermo Fisher Scientific, for sponsoring today's webinar. Questions submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 1 year until May 14th, 2026. Laboratories will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.